Okay, good morning, everyone. It's nice to see you out in the virtual world today. I hope you are doing well and your families are safe. Okay, let's see. If you can hear me okay, please type yes into the chat window. Okay, thank you very much. All right, gang. Um, just can you please tell me, did you receive the study guide for the final exam on this past Monday's email? Please type yes into the chat window if you received the study guide. Okay, good. All right. I just want to be sure that you are receiving the information that I'm trying to get out to you. Okay, um, so yes, we are rapidly coming towards an end here. Your final exam, as you know at this point, is Monday, May 11th, and you now have your study guide in your hands. So hopefully that'll allow you to focus your study efforts. And you know, we do have lab exam 3 coming up this coming Monday, May 4th. Okay. Are there any general questions at this moment about the course schedule, about what you can expect moving towards the end? Okay, so a couple questions coming through the chat window. Is there a certain time we need to take the test? Which test are you asking about, Jennifer? The lab exam or the final exam? Okay, for the final exam, even though the school says that you should technically be taking it at 8 a.m., I'm going to leave the exam open all day long as I typically do. In other words, as long as you take your final exam on Monday, May 11th between 7 a.m. and midnight, that will be fine. Okay, so you may take it anytime between 7 a.m. and midnight on Monday, May 11th. Okay, Allie, the final exam is Monday, May 11th. Okay. All right, any other questions? Okay, not seeing anything. What we're going to do is get back in and cover the second part of chapter 15 here. So we're going to continue our conversation with chapter 15 and get as far as we can. And then coming next week, we will be covering chapter 16 as best we can. Okay, so please stay focused. You all can do this. I have confidence in you. Okay, and you can finish strong in this semester. Let's go to the Word document. And okay, let's see here. I have the chat window up if you need to get a hold of me while I'm talking. And at this moment, you should see a Word document up. And I have placed on the very front of our Word document the past couple notes we took from last class on Monday. Okay, so recall that we started last class talking about similarities and differences between the somatic nervous system and the peripheral nervous systems. And then we went into a practical example where we're sitting at a 4D movie theater and we're getting scared. Okay, we're getting scared. And we're trying to connect that to some of the content of chapter 15. Okay, so we're in this 4D movie theater and you see scary images, and those scary images are being communicated from the retina of your eyes into the cerebral cortex via the optic nerve, which you know formally is cranial nerve number 2. Okay, you can see that, you know, there are 5 chapters of nervous system content in the textbook, and we don't have time to cover every chapter in detail. So I cover 3 chapters with you in lecture and then assign homework through the lab portion of the course, where hopefully you get comfortable with some of the other aspects of the nervous system. I do know that one of the homeworks I've assigned goes over the cranial nerves, right? So hopefully cranial nerve 2 is not a completely foreign idea to you. All right, so we're getting scared. The eyes send information to the cerebral cortex. The sounds of the scary video are being sent into the temporal lobe of the cerebral cortex via the vestibulocochlear nerve, which is cranial nerve 8. Okay. And then your nose, right? In these 4D movies, sometimes they throw out aromas and odors and fragrances. Well, they could throw out a gross odor, kind of a repugnant odor, and your olfactory receptors in your nose would send information also to the cerebral cortex via the olfactory nerve, which is cranial nerve number 1. While all this is happening, there's special tubes on the back of your chairs as you sit at the 4D theater, and they're blowing out cold air onto the back of your neck. So you feel the coldness of the air through thermoreceptors embedded in the dermal layer of your skin, and you can literally feel the hair on the back of your neck moving from the cold air that's pushing on it. That's because there are hair root plexuses embedded around each hair follicle. Okay, so that's basically where we left off with our conversation of being scared. Okay, so notice that the input, right? The input to the central nervous system is all letter D. We're experiencing things basically at the surface of our body. Next, all those neurons from the eyes, from the ears, from the nose, from the dermal layer of your skin are transmitting information up the spinal cord and up into the brain. This is where the integration portion of our conversation continues. So we're in letter E. The integration aspect of this experience, this 4D scary movie experience, is happening at this time. So number 1, networks of neurons communicate with each other through interneurons, which act as bridges between one region of the central nervous system and another. Okay, so let's continue here. Roman numeral number 2. The whole purpose, folks, of integration is to allow your brain to process the stimuli, right? You're getting sent multiple stimuli from multiple sensory organs into the brain. So the purpose of integration is to allow the brain to process the stimuli so you can perceive, understand, interpret the stimulus, and then communicate that information to other regions of the brain that act as command and control centers so that you can potentially respond to the stimulus that your body is experiencing. Let's see if we can get that down in your notes. The purpose of integration. Okay, now if you've had any math classes, particularly higher math like calculus, you know there's a topic in calculus called integration. That's not what we're talking about at this time. Okay, that's not what we're talking about. The purpose of integration is to process stimuli so you can perceive, or if you prefer, understand, or if you prefer interpret, okay, the stimulus, okay, such as the cold air on the back of your neck, and to communicate, okay, to communicate via the interneurons, right? There's going to be these intermediary middlemen, right? You know, if you work for a large company, very likely if you're just at the entry level of the company, you probably can't talk to the company president, right? You got to talk to your manager. So there's always a middleman, a middlewoman. There's a person, there's a go-between. That is the function and purpose of the interneurons. And to communicate via the interneurons to other regions of the brain that are so-called command and control centers capable of producing a response from the stimuli. Okay. So let's think about it. You know, let's say that you're feeling the cold air on the back of your neck. So that means neurons embedded in the dermal layer of the skin on the back of the neck are detecting that difference in temperature change at the surface of your body. That's creating an electrical impulse, the action potential we talked about from chapter 12, that is in fact spreading up into the brain. Now, wherever those signals go, that information is then going to be relayed by a middleman, by an interneuron, to a command and control center. And probably the most well understood and most common command and control center we talk about in the brain is actually the hypothalamus. So Roman numeral 3, the master command and control center, linking the nervous system to the endocrine system, okay, is the hypothalamus. Okay. Remember, everything we're talking about in our scary scenario here at the 4D movie theater, responses to the stimuli are going to be coordinated both through the nervous system and the endocrine system, okay? Okay, so let's take a moment here. You know, this is a challenging topic to learn because there's so much content, and basically we don't have all the content we need to understand this in a single chapter, right? Okay, we talked about the nerve impulse in chapter 12, so we have to go back there to talk about details of the nerve impulse. I'm not going to do that at this time, but in the first section of our notes here, the input section, I reference the cerebral cortex several times, okay? Number 1, number 2, number 3, these things are referenced several times. So let me ask you folks, okay, in the so-called virtual audience, what figure in chapter 14 shows you the lobes of the cerebral cortex? Can you find that for me, please? If you have your textbook available, I hope you do. Can you tell me what figure in chapter 14 shows you the lobes of the cerebral cortex? See if you can track that down, gang. Okay, there's actually a couple of figures that you could get this information from. Who's going to get to it first? Okay, from the first part of our scenario, we need to know where the occipital lobe of the cerebral cortex is. Okay, Mosena, thank you very much. Exactly. Figure 1411 on page 497 is one correct possibility, right? Those, on page 497, you do see the surface anatomy of the brain, and it does point out the various lobes of the cerebral cortex. Remember, the cerebral cortex is the top surface area of the brain. Okay. All right. Let's see here. Okay. So I'm looking at, yeah, Pamela, figure 14.8 does show you the parts of the cerebellum, I believe. And so that's not the same thing as the cerebrum, okay? So be careful about that because they're both C words. So if you look on page 497 or page 501, either of those pictures is going to show you the anatomy of the cerebral cortex. And so just put an eyeball on where the occipital lobe is and where the temporal lobe is. You can basically see we're talking about the bottom portions of your brain, okay? So when your eyes send information by the optic nerve, that information's literally going into the neurons of the occipital lobe. If you look on page 497, that would be the green colored area, the green colored area. When you receive information from your ears, and that information goes along the vestibulocochlear nerve, that's going to be transmitted into the temporal lobe, which you can see is like a light pinkish purple color. Okay, so very good, folks. You found those two lobes. Now, tell me, what figures in chapter 14 show you where the hypothalamus is? Okay, what figures in chapter 14 show you the hypothalamus? (1410), okay. Yes, thank you very much. Thank you, Ashley. That's correct, okay? So now you can see where the hypothalamus actually is, okay? Actually, what they're really showing you on figure 1410 is the various clusters of neuronal cells within the hypothalamus, right? The paraventricular nucleus, the lateral preoptic nucleus. Of fame is the suprachiasmatic nucleus there, which contains neurons involved in your circadian rhythm and your biological clock. All of those make up the hypothalamus, okay? So you can kind of get a reference of where the hypothalamus is in relation to the occipital and temporal lobes by looking at that tiny little brain to the left on figure 1410 there. So what we're saying at this moment is that those sensory stimuli that you're experiencing at the surface of your brain, excuse me, at the surface of your body from the 4D scary movie, those stimuli are going into the occipital and temporal lobes. And then those neurons, the axons of those neurons are synapsing with dendrites of interneurons, which then take that information and transmit it into the neurons of the hypothalamus, okay? That's kind of the picture that I want you to appreciate, okay? So let's put that down, okay? The axons of neurons transmitting stimulus information into the occipital and temporal lobes of the brain are synapsing, okay? Synapsing, meaning they are connecting with. Now, you know, if you've been reading your book carefully, particularly from chapter 12, that there's two kinds of synapses. There are biochemical synapses, which always involve the release of a neurotransmitter. And then there's also lesser known, but nonetheless, electrical synapses where you have cells merging their plasma membranes together, okay? So at this point, we're primarily talking about the chemical synapse, okay? So these axons are synapsing with dendrites of interneurons. Okay. So, let me ask you well, I guess maybe we'll put this into your notes. Here's a question for you. Okay, here's a question for you. When the presynaptic neuron, okay, so these are the neurons that are sending these scary stimuli into the brain. Okay, these are the neurons sending the scary stimuli into the brain. When the presynaptic neurons activate dendrites on the interneurons, what is the nature of the interneuron's first response? In other words, what does the interneuron do when it is stimulated by the presynaptic neuron? Okay, when the presynaptic neurons activate dendrites on the interneurons, okay, the dendrites on the interneurons are going to be the postsynaptic cell. Okay, what is the nature of the interneuron's first response? Okay, let me kind of guide you here. There's going to be an electrical signal created in that interneuron. What is the nature of that electrical signal? What kind of electrical signal is it? Let me type that into the chat window. What kind of electrical signal is created in the dendrites of the neurons when they are stimulated by the presynaptic axonal signals? I'm trying to pull something out of you folks. Can you answer that for me? What kind of electrical signal is created in the dendrites of the neurons, interneurons? Okay, so Lindsay, you say a nerve impulse. Can you be more specific, Lindsay? Pam says an action potential. Okay, that is not correct. Okay, Lindsay says a graded potential. That is correct. Okay, folks, remember there are two kinds of electrical signals in the nervous system. There are the graded potentials and there are the action potentials. When a presynaptic neuron activates an electrical signal in the dendrite or even in the cell body of the postsynaptic neuron, that signal is graded. That signal is a graded potential, which means it has varying amplitude. Okay. A graded potential is created. So the answer to the question, when the presynaptic neurons activate dendrites on the interneurons, where the interneurons would be defined as our postsynaptic cell, what is the nature of the interneuron's first response? That the answer is that a graded potential is created. Okay. Okay. Next question. Folks, I'm trying to connect the content of chapter 12 to your content here in chapter 15, right? We're trying to do something really hard, which is to not overly compartmentalize the chapters. We want to see the connection between the content of the two chapters. Okay? What must happen for an action potential to be triggered in the interneuron? What must happen for an action potential to be triggered in the interneuron? Okay, so let's back up here, right? The stimuli we're receiving at the surface of our body through our eyes and ears, that signal is being sent up the neurons, up the axon, which is then synapsing with a postsynaptic cell, which is the interneuron. The first thing that happens is that the interneuron generates a graded potential. So we understand that. Now I'm asking you the next question. What must happen for an action potential to be triggered in the interneuron? Can you answer that? Folks, what I'm trying to get you to see is what has to happen for the electrical signal, which is the graded potential, to transition into an action potential. What has to happen? Okay, Mosana, you says you have to get to threshold. Okay, I like that answer. That really helps us out here. Can you define threshold with a number for me? So folks, Mosana said threshold. Threshold would not be plus 30, no. Okay, I'm glad. I'm very happy to see that you're starting to put some numbers on this, but nobody's put the right number on it yet. Pam, you said minus 70. Minus 70 is the resting membrane potential of the neuron. So that's not the proper voltage. What's the, there we go, ladies. Fantastic. Okay, minus 55, right? Minus 55 millivolts is the threshold potential. Fantastic. That's it. Okay. So one more question from your annoying teacher. For the interneuron to reach that threshold potential, are we going to have to depolarize or hyperpolarize the neuron? Depolarize. Very good. Very good, ladies. Okay, let's put this down. Well, actually, one more annoying question from your teacher. What anatomical region of the neuron has to depolarize to minus 55 millivolts? No, not the synaptic end bulb. That's where you're going to release neurotransmitter. That's not correct, Lindsay. Okay, Messina, you say the axon. Can you be even more specific? Not the dendrites. No, not dendrites. It is the axon. Be even more specific, Messina. Not the axon terminal. No, the axon terminal. There it is. The axon hillock, right? The axon hillock. That's exactly what I'm fishing for here. Okay. So, okay, ladies, if you want to take a look at that, let's see here. I'm just looking at who's online at this time. I don't think we have any guys online at this time. So, okay, guess we scared them all away. Take a look at page 407. figure 12.2. Okay, there you can see that, okay, so the electrical signal of the presynaptic neuron is getting regenerated in the dendritic spines of the interneurons. The dendritic spines of the interneurons, which are generating graded potentials. Those graded potentials are going to spread along the plasma membrane towards the axon. And when you get to the axon hillock, okay, notice where that phrase axon hillock is pointed out on figure 12.2. When you reach a depolarization significant enough to achieve minus 55 millivolts, a difference of electrical charge across the membrane of minus 55 millivolts, then you will trigger an action potential down the rest of the axon. And that action potential, as you've read, is an all or none event. So once we start it at the axon hillock, once we start it, it's like pulling a trigger on a gun. You fire the bullet and you cannot unfire it. It's like you crack an egg, you cannot uncrack an egg. So once minus 55 millivolts is triggered at the axon hillock, bang, we generate an action potential. It propagates down the axon. Okay? All right. Folks, are you with me? Are you okay? Is this making sense? I'm asking you, is this making sense? All right, folks. Let me know how I can help you. Okay, we want to deepen our understanding. We don't want to stay at a surface level here, right? You folks are capable of more than that. Okay, so let me answer this question in your notes. What must happen for an action potential to be triggered in the neuron? The plasma membrane at the axon hillock must depolarize to minus 55 millivolts, okay, which is the threshold potential. Okay, let's keep going here because I don't want to, I want to be able to get my notes into you today. Okay. All right, so everything we've talked about in letter E is now integration. Okay, so the signal is being processed in the brain, and for that to happen, the presynaptic neurons of the sensory nervous system are synapsing with the interneurons in the occipital lobe and temporal lobes of your brain. A graded potential starts in the dendrites, it then propagates to the axon hillock, where we depolarize it to threshold potential of minus 55 millivolts. We trigger an action potential that goes down the axons of the interneurons, leading to some sort of command and control center, which the one that I'm going to talk about and expect you to know is the hypothalamus of the brain. So interneurons are sending their action potentials from the occipital and temporal lobes into neurons of the hypothalamus. Okay, next step. Next step is the output. Okay, so the output is going to involve the peripheral nervous system, and if we're being honest, also the endocrine system too. Peripheral nervous system and the endocrine nervous system as well. Okay, first thing to note, okay? So all of this integration is happening in various areas of the brain. There's all these synapsing of sensory neurons to interneurons, and all this information is being sent to a command and control center like the hypothalamus, which is making the decision that says, oh, I'm experiencing something scary right now. This is fear happening that I'm experiencing. Okay, so the hypothalamus is making the decision that all the stimuli are scary stimuli. Okay. Because integration in multiple areas of your brain lead to the perception of fear, right? I mean, you can see something, but if you don't perceive it as a threat, then it's not a threat, right? So your brain has to be thinking, oh, what I'm seeing, what I'm hearing, what I'm smelling, what I'm feeling on my skin are things that make me uncomfortable. So that generates a fear sensation in your brain. Okay? The hypothalamus has received multiple electrical signals via the interneurons that makes the decision that, yes, these are fear stimuli. Okay. So the hypothalamus interprets these action potentials as fear. Okay. And so the hypothalamus, being a command and control center, it's going to generate responses in your body. Okay. So we have multiple responses that we need to talk about. Okay. Excuse me a second while I get to a certain page that I need here. Okay. All right. So I'm going to code this as response number one. Okay, response number one, because there's going to be multiple responses that your hypothalamus is responsible for. Okay. So the hypothalamus responds by sending nerve impulses down axons that are part of the sympathetic division of the autonomic nervous system. Okay. So let's do this. Hypothalamus response number one. Hypothalamus sends nerve impulses down axons that are part of the sympathetic division of the autonomic nervous system. Okay, so for sympathetic, should I be thinking fight or flight or rest and digest? What should I be thinking? Fight or flight, or rest and digest? Type that into the chat window if you know the answer there. Okay, we're talking about the sympathetic division here. So am I thinking fight or, okay, Ashley, thank you very much. It is fight or flight. Okay, let's continue here. Okay, axons, axons from the hypothalamus, okay, go down through the spinal cord. Axons leave the spinal cord at thoracic vertebrae T10, okay, thoracic vertebrae T10. When they synapse, or where they synapse, with chromaffin cells embedded in the adrenal medulla of the adrenal gland located on top of your kidneys. Okay, so let's see here. So if you want to visualize this, you can visualize this in figure 15.2, page 530. Figure 15.2, page 530. Go down on that image of the spinal column there and locate thoracic vertebrae T10 and trace that blue line out till you get to the adrenal gland. Okay, so you notice that it splits off a little bit there, but it goes through, well, let me ask you, what ganglion does the axon from T10 go through as it passes the signal to the adrenal gland? Can you answer that? What ganglion processes the signal from the axon coming out of T10 as it transmits to the chromaffin cells of the adrenal gland? What ganglion? Who's going to be first to answer that? Do you see what I'm talking about, gals? Okay, Alexia says celiac, and Ashley says the renal ganglion. Well, it is the celiac. Alexia, you are correct. The celiac ganglion, okay? So I'm pointing this out, folks, because you noticed if you read the study guide for the final exam, I'm going to be expecting you to create flowcharts where you explain things to me. So you have to be able to look at the pictures in your textbook and say, okay, the axon coming off of this vertebral region of the spinal cord goes through this ganglion to transmit a signal to a given organ. This is the kind of detail you need to focus on as you prepare an answer for your final exam. So, Alexia, good job. That is correct. Okay. Questions right now? Do you all see now that we're saying it's the celiac? Does that make sense to you? Do you see that that's correct? The celiac ganglion. Okay. Okay. All right. Good. All right. Let's continue here. So, we have the axons leaving the spinal cord at vertebra T10, where they synapse with chromaffin cells embedded in the adrenal medulla, located on top of your kidneys. Okay. Then these chromaffin cells release acetylcholine. Okay. So there's a release of acetylcholine. Excuse me, there's a release from the preganglionic neurons of acetylcholine, which actually stimulates the chromaffin cells then to release the adrenaline, the epinephrine and norepinephrine. So there's a release of the acetylcholine by the sympathetic preganglionic neurons, which stimulates the chromaffin cells in the adrenal glands to release epinephrine and norepinephrine. Release of acetylcholine by the sympathetic preganglionic neurons, stimulates the chromaffin cells in the adrenal glands to release epinephrine and norepinephrine. Okay, so the hypothalamus, the command and control center of the brain, received information via the interneurons. It made a decision that there was fear that you were experiencing, and so the hypothalamus decides that it needs to get the adrenal gland to release adrenaline. To do that, it sends an action potential down the axon of the spinal cord, which leaves the spinal column at vertebra T10. It goes over and it synapses with a preganglionic neuron in the celiac ganglion, which releases acetylcholine, okay? That continues to signal down to the chromaffin cells, which then respond by releasing epinephrine. Okay, the next part. Your body responds to these hormones, epinephrine and norepinephrine, right? These stimulatory hormones here. Your body responds to that by increasing heart rate, and converting glycogen back to glucose. Okay, now the actual details of this is, we're skipping a lot of important steps. Pamela, your question on the chat window, are epinephrine and norepinephrine released by the adrenal glands? Yes, they are. That is released by the adrenal glands. Okay. Okay, so your body responds to these hormones by increasing heart rate and converting glycogen back to glucose. The chemical processes of doing that, we're skipping the details of that. We're not going into that kind of weedy conversation at this point. Okay, converting glycogen back to glucose to increase blood flow, to increase blood glucose levels. To prepare for conflict. Okay, we want our blood sugar level to go up so that there's plenty of sugar to power muscle contraction, since we're scared and we might want to contract our leg muscles to run away from the stimulus. Okay, so take note here that heart rate and the force of heart contraction are influenced by endocrine signals that are triggered by nervous system activation. Okay, right? The nervous system via the action potentials and also some neurotransmitters is stimulating the release of the hormones epinephrine and norepinephrine into the bloodstream, which then elicits a whole nother series of effects that we're choosing right now not to explain in great detail, because you're going to get the endocrine system in AMP2. All right. Questions, folks? Okay, so the first response can be summarized. I'm moving my cursor up to hypothalamus response number 1 is triggering the release of epinephrine and norepinephrine. Okay, that's the first response, triggering the release of epinephrine and norepinephrine from the adrenal glands. Okay, any questions? Okay, let's go to the next bullet point. it will be hypothalamus response number 2. Hypothalamus response number 2, which is going to be, I'll give you the punchline at this point. The second hypothalamus response is going to trigger a reduction or a decrease in saliva production. I don't know if you've ever noticed when you're fearful in any way, but typically you experience dry mouth, don't you? Your mouth gets really, really dry. Think about maybe the last time you had to give a speech in front of people and you were nervous about it. Your mouth probably got really dry, okay? Trigger a decrease in saliva production. Okay, this is all associated with being scared. Okay. So the hypothalamus sends nerve impulses down other axons of the sympathetic nervous system. Okay, they're not going to be sent down the exact same ones of response number 1, because obviously we need the signal to go to a different anatomical location of your body. So nerve impulses are sent down other axons of the sympathetic nervous system that eventually synapse with cells in the superior cervical ganglion. The superior cervical ganglion, which is on page 530, figure 15.2. See if you can identify the superior cervical ganglion. Try to put some eyes on that at this time. Okay, so that eventually synapse with cells in the superior cervical ganglion. Okay. Here, the preganglionic neurons, release acetylcholine. Okay. It's kind of neat to see this because, you know, many times in beginning human bio courses, you talk about acetylcholine and you always talk about it in terms of a neuromuscular junction of how acetylcholine is released to stimulate muscle contraction. And that's correct and that's good stuff. But please know that acetylcholine is used for much, much more than neuromuscular junction activity. Here are the preganglionic neurons release acetylcholine to stimulate the postganglionic neurons. Okay. The postganglionic neurons communicate by the release of norepinephrine. Postganglionic neurons communicate through or by the release of norepinephrine to arterioles, okay, arterioles, so medium-sized arteries contained within the salivary glands, causing those arterioles, the smooth muscle of those arterioles to constrict. Okay. If the smooth muscle of the arterioles contract to constrict or narrow the arterioles, that means you're going to have less blood plasma leaving that area of your anatomy, which means you have less liquid available to actually form saliva. So it is the constriction of the arterioles. It's the constriction, it's the decrease in their diameter caused by the contraction of the smooth muscle that decreases saliva production. In other words, this gives you dry mouth. Okay. Question, ladies. Okay. So on your study guide for the final exam, I challenged you with 2 alternative scenarios to design flowcharts. And I'm giving you a hint with these notes of the kind of information I would like in those flowcharts. Okay, any questions? All right. Okay, so we've done hypothalamus response number 1, right? We've done that. We've done that. We've done hypothalamus response number 2. And now what I want to do is talk about some additional responses that occur in your body after the sensation of fear has passed, after the danger has passed. Okay, so next. After the danger or threat has passed, the hypothalamus triggers additional responses. Okay, so after the danger has passed, the hypothalamus triggers additional responses. So we're going to go with hypothalamus response number 3. Number 5 here. Hypothalamus response number 3. Okay. All right. Just to see how the body resolves issues. Okay. Well, okay, the hypothalamus after the danger has passed. So, let's be honest with you. At this point, a lot of things have happened. Maybe the movie you're watching in the 4D theater is over, right? You're not seeing those scary images anymore. Nobody's blowing cold air on the back of your neck anymore, okay? So the stimuli for the fear production has stopped, and the hypothalamus is aware of that. So the hypothalamus will send nerve impulses down axons that are part of the system that has to cause you to slow down, to relax, to rest. So what division am I going to be in at this moment? What division of the autonomic nervous system am I going to be in? This axon that I need to communicate in. Okay, thank you, Ashley. It's going to be in the parasympathetic division. So the hypothalamus is going to send nerve impulses down axons that are part of the parasympathetic division of the autonomic nervous system. Okay, and I'm going to have to move a little bit quicker, gals, because I got still a lot of notes I want to get into you here. Okay, so axons leave the brainstem above cervical vertebrae C1. as part of the facial nerve. Okay, so that's cranial nerve 7. So these axons leave the brainstem above C1 as part of the facial nerve. Remember, a nerve is a bundle of axons. A nerve is a bundle of axons. These axons synapse with other neuron cell bodies that are in the submandibular ganglion. Okay, that are in the submandibular ganglion. So remember, we're looking at the parasympathetic division. So for the parasympathetic division, am I looking at figure 15.2 or figure 15.3? Am I looking at figure 15.2 or 15.3? What do you say, ladies? Should you be looking for the submandibular ganglion in figure 15.2 or 15.3? Okay, thank you, Ashley. 15.3, right? So we've got two fairly big, complicated pictures here. Make sure you're not swimming down the wrong path, so to speak, that you're looking in the right area. 15.3 is correct. Okay, do you see the submandibular ganglion there? Okay, you notice where it is, and you notice how it's communicating out to the salivary glands. Okay, let me finish these notes for you. Okay, within the submandibular ganglion, parasympathetic, preganglionic neurons release acetylcholine, which activate parasympathetic, postganglionic neurons to release their own acetylcholine, which causes the arterioles in the salivary glands to dilate, promoting saliva production. Okay, in other words, we are relieving our dry mouth. It's going to get better. Okay, let me just read that note to make sure that it's correct. Within the submandibular ganglion, parasympathetic, preganglionic neurons release acetylcholine, which activate parasympathetic postganglionic neurons to release acetylcholine, which causes the arterioles in the salivary glands to dilate, to open up, to increase the blood flow, which supports an increase in saliva production. We are relieving our dry mouth. Okay. All right, last thing here, then we can move on to another topic. I know we're really digging through the mud at this point. Let's see here. This is going to be hypothalamus response number 4. Okay, so we have hypothalamus response 3 and hypothalamus response 4 occurring after the threat has passed. at the same time as response 3, right? I mean, I can only talk about one thing at a time, but your body is capable of rapidly multitasking to engage different parts of the parasympathetic division simultaneously. So while we are relieving our dry mouth scenario, we can also be relaxing our heart rate. So at the same time, nerve impulses are sent to the, okay, you tell me, nerve impulses are going to pass through what ganglion so that we can relax the heart? What ganglion is going to receive nerve impulses in order to relax the heart? What do you think, gals? Okay, thank you. Thank you very much. Do you see that it is in fact the otic ganglion? The otic ganglion. Okay. Um, okay. Lindsay, I noticed you put in the middle cervical. Notice the middle cervical ganglion is part of the sympathetic division, and that's all involved about accelerating the heart. We don't want to accelerate the heart because the fear has passed. We want to calm down. So we need to be looking at the otic ganglion of the parasympathetic division on page 531. Okay, at the same time, nerve impulses are sent to the otic ganglion, where acetylcholine is released to stimulate postganglionic neurons to communicate with the heart. Okay, also via acetylcholine to slow heart rate and reduce the force of heart contraction. Okay. All right, gang. So, certainly we did not talk about everything we could have talked about here. There's still more things that happen when you feel fear. And there's more things that happen to calm you down after the fear leaves you. But I'm hoping that I gave you enough to give a sense of how the nervous system is communicating along the sympathetic and parasympathetic divisions. Okay, questions at this time? I know this is pretty heavy stuff, but this is what's going to separate you as an educated person from someone that's not educated. This is what's going to separate you as being someone that can be a successful nurse down the road, that you know details that other people don't, that you observe things that other people don't. That's really what your education is about. And so you have to go through some growing pains to get there. Okay, let's continue here. I want to talk to you a little bit about the neurotransmitters and receptors that are part of the autonomic nervous system. Autonomic nervous system, neurotransmitters and receptors. Just make a couple of general comments here about things that I'd like you to be familiar with. Remember, we've said this already, remember that the same neurotransmitter can create different responses in different locations of the body based on differences in the receptors of the target tissue. Okay. Number 2. Okay. In the notes we've given you, we've talked primarily about 2 categories of neurotransmitter. We talked a lot about acetylcholine. Well, acetylcholine is going to be released by neurons which are described as cholinergic neurons, right? Cholinergic neurons release acetylcholine. And we also talked a little bit about norepinephrine and epinephrine. Well, norepinephrine and epinephrine, which are 2 different neurotransmitters, are released by adrenergic or adrenaline-based neurons. Okay, so we need to put this in your notes. Cholinergic neurons release acetylcholine, right? Hopefully you can see the commonality here, right? Cholinergic and acetylcholine. Okay, I'm underlining choline in both of those terms there, right? Cholinergic neurons release acetylcholine, which then diffuses to their target receptors. Okay, cholinergic receptors bind to acetylcholine, and they come in 2 varieties, right? We can have 2 different responses to acetylcholine based on 2 different types of receptors. Cholinergic receptors bind acetylcholine and come in 2 varieties. We have nicotinic receptors, okay? Nicotinic receptors. Let's see here. So you can look at the various kinds of neurons and some of the information about these receptors on page 538 and 539 of your textbook. That's what I'm talking about, pages 538 and 539. Nicotinic receptors, they're named this way because nicotine can also bind to these receptors just like acetylcholine, okay? In other words, sometimes in Mother Nature, there are multiple substances that have enough of an identical shape that they can bind to a common receptor. So nicotinic receptors are named because nicotine can also bind to these receptors just like acetylcholine, okay? This means nicotine is an fill in the blank. Can you fill in the blank? We're almost, God doggone it, we are out of time. Let's finish this and then I'll let you go. Folks, is coming through the chat window, is nicotine an agonist or antagonist? Agonist, very good. This very, very good. That's exactly right. Okay, this means nicotine is an agonist for these receptors. Okay, in other words, they can bind and activate the receptor just like acetylcholine. Okay, all right. I'm a little bit over, so I think I will stop here. So I have maybe 10, maybe 10, 12 minutes worth of notes that I need to finish with you from chapter 15 on Monday. And then we'll be hitting chapter 16 hard. Okay. So do you have any questions at this time? Yes, Jennifer, I will share these notes with you this afternoon. Anything else? Okay, then I'm going to stop sharing my window here. And I'm going to take about a 15-minute break. I will be back online, and if anybody is interested, I will give you the answers for your homework that is technically due from your syllabus. I'll give you that. And I will also give you a small practice quiz for the upcoming lab exam on Monday. Okay? So, do you have any questions before we take a small break? Okay. Then, is this practice quiz the same as Monday? Yes, Jennifer, it will be the same questions. Okay. Okay. Gals, if I don't see you, please continue to stay safe. You know, if you've been paying attention to the news media, that coronavirus is definitely spiking in our geographic region of the state right now. It's pretty bad out there in our region. So be very careful when you go out. Make sure you wear a protective covering when you're in stores. Be safe, and I'll see some of you in 15 minutes for the practice and homework information. Otherwise, have a nice weekend, and I will see you soon. Okay, welcome back, Lindsay and Pamela. Gals, are you with me right now? Please type into the chat window if you're actually online. Okay. All right, so let's go over the homework for lab exercise 18. You can correct your own work. We'll take reviewing your knowledge first on page 293. Page 293, reviewing your knowledge. Letter A, the connective tissue coverings. Okay, number 1 is the endoneurium. Number 2 is the perineurium. 3 is the epineurium. For section B, the rami of the spinal nerves. Number 1 is the posterior ramus. 2 is the rami communicantes, Number 3 is the anterior ramus. Letter C for the major nerves of the nerve plexuses. Number 1 is the lumbar. Number 2 is sacral. Number 3 is cervical. 4 is brachial. 5 is brachial. 6 is sacral. 7 is lumbar. 8 is brachial. 9 is sacral. And 10 is sacral. So do you need any of that repeated from page 293? Okay. Number 2 from section B, that is rami communicantes. I'll type that into the chat window so you know how it's spelled. Rami communicantes. Anything else? Okay, let's turn the page. Page 294, letter D, the spinal nerves. Number 1 is tibial. 2 is the common fibular, which you might have said as common perineal. It's the same thing. Number 3 is 31. 4 is sciatic. 5 is the number 1. 6 is motor and sensory. 7 is 12. 8 is femoral. 9 is the number 5. 10 is also 5. 11 is motor and sensory. 12 is the number 8. 13 is axillary. 14 is radial. 15 is musculocutaneous. 16 is phrenic. 17 is ulnar. 18 is median. Do you need anything from section D repeated? Okay, let's go to page 295. Letter E, the anatomy review. We'll take a look at figure 18.7. Number 1 is the epineurium. 2 is a spinal nerve. 3 is the perineurium. 4 is fascicle. 5 is endoneurium. 6 is axon. Looking at figure 18.8. Figure 18.8, number 1 is axillary. 2 is musculocutaneous. 3 is median. 4 is radial, 5 is ulnar, and 6 is phrenic. Okay. Do you need any of that repeated? Okay, let's go to page 296 and look at figure 18.9. Okay, number 1 is obturator. Obturator. Number 2 is pudental. P-U-D-E-N-D-A-L. Pudental. 3 is femoral. 4 is sciatic. 5 is tibial. And 6 is the common fibular. Okay. All right. Let's go to page 297, using your knowledge. Okay, page 297. Number 1 is the median nerve. 2 is the radial nerve. 3 is the sciatic nerve. Okay. Number 4 is the ulnar nerve, which affects the medial part of the hand, the medial part of digit number 3, and all parts of digits 4 and 5. So number 4 is the ulnar nerve. Number 5 is C8. C8. Number 6 is the obturator nerve. 7 is the common fibular nerve. 8 is the sciatic nerve. 9 is the axillary nerve. And 10 is the ulnar nerve. Gals, do you need anything from page 297 repeated? Okay. All right. So that was exercise 18. Let's go on to exercise 20. Exercise 20, reviewing your knowledge, page 327. Page 327. Okay, we'll take a look at letter A for the brain structures. Here we go. Number 1 is the thalamus. 2 is the hypothalamus. 3 is the pineal gland. 4 is the diencephalon. 5 is the midbrain. 6 is pons. 7 is medulla oblongata. 8 is the brain stem. 9 is the cerebellum. 10 is the spinal cord. 11 is cerebrum. And 12 is the pituitary gland. Do you need any of that first part repeated? Okay, let's go to picture B on page 327. Number 13 is the cerebrum. 14 is the olfactory bulb. 15 is the olfactory tract. 16 is the pituitary gland. 17 is the optic tract. 18 is the cerebral peduncle of the midbrain. 19 is pons. 20 is the medulla oblongata. 21 is the spinal cord. 22 is the cerebellum. Okay, any part of that that you need repeated? Okay, let's turn the page, page 328, section B, functions of the brain regions. For number 1, on page 328, part B, number 1, is the medulla oblongata. Number 2 is the cerebellum. 3 is the pineal gland. 4 is the hypothalamus. 5 is the cerebral cortex. 6 is the pons. 7 is the thalamus. 8 is the corpora quadrigemina of the midbrain. Okay, that's quite a mouthful. The corpora quadrigemina of the midbrain is number 8. Number 9 is the basal nuclei, and 10 is the corpus callosum. Any of that you need repeated? Okay. All right. So let's move to section C, where we're talking about flow of the cerebrospinal fluid. Number 1 would be the choroid plexus. Number 2, the lateral ventricles. 3, the third ventricle. 4, the fourth ventricle. 5, the ependymal cells. 6, the septum pellucidum. 7, the third ventricle. 8, the cerebral aqueduct. 9, the subarachnoid. 10, the central canal. 11 is subarachnoid. 12 is the arachnoid villi. And 13 is the superior sagittal sinus. Okay, do you need any of that repeated? Okay, let's finish our work with using your knowledge, page 329. We can look at section A. Here we have an MRI scan of the human brain. Okay, page 329. Number 1 is the corpus callosum. 2 is the thalamus. 3 is the midbrain. 4 is the cerebellum. And 5 is the pons. Turning the page to page 320, we continue with section B, the transverse section of a human brain. Number 6 is the falx cerebri. That's F-A-L-X, second word, cerebri. Falx cerebri is number 6. Number 7 is the corpus callosum. 8 are your lateral ventricles. 9 is the septum pellucidum. 10 is the choroid plexus. Okay, page 331, section C, the sagittal section of the human brain. Number 11 is the lateral ventricle. 12 is the tentorium cerebelli. 13 is the cerebellar peduncle. 14 is the dura mater. 15 is the corpus callosum. Do you need any of that repeated? Okay, let's turn to page 322. The last section was a small part on brain injury. Okay, so 16 is letter D, a loss of vision. 17 is letter B, a loss of equilibrium. Let's see here. Pam, I see your question, and I'll come back to that. We're on section D. Once again, 16 is D, loss of vision. 17 is B, loss of equilibrium. 18 is A cessation of breathing. 19 is E, a loss of pain localization in the shoulder. 20 is C, a loss of the use of the left arm. Okay, Pam, in the chat window, you asked for number 9 on part B. That is the septum pellucidum, okay, which I will type into the chat window so you can see the spelling. Septum pellucidum. Okay. Ladies, do you need anything else repeated? Okay. All right, then why don't I give you a small practice quiz at this time. I'm going to show you 4 practice images that are going to be on this coming Monday's lab exam. So just a moment while I get my computer files up here. One moment. Let's see here. Okay. Gals, if you are ready to start the practice quiz, please type in ready into the chat window. Okay, Pamela and Lindsay, I'm going to start sharing my screen with you, and I'll give you 45 seconds on each of these practice images. Okay, one moment. Okay, for the first practice question, please identify this structure in the brain. 45 seconds, please begin. Okay, that was definitely 45 seconds. Let me go to the next one. For practice question number 2, please identify this structure in the brain. Here it is. Okay, 45 seconds, go ahead. Okay, that's 45 seconds. Let's go to the third practice question. For the third practice question, you're going to be identifying a structure in the eye. Okay, so here it is. 45 seconds, please begin. That's 45 seconds. Let's move to the fourth and final practice question. Fourth and final practice question is going to be this structure here. Okay, you see a green arrow and it's pointing to something that looks like a blue fiber here on both sides of the brain stem. Okay, 45 seconds, please begin. And that is 45 seconds. Okay, so let's see here. Let's see how you did, ladies. For the first practice image, which is this image here, please type into the chat window if you had a guess. Okay, very good, gals. That is, in fact, the amygdala. That is the amygdala. Well done. Okay. For the second practice image, okay, did you have a guess for this one? Okay, so I'm not seeing anything, so I suspect maybe this was a little bit harder, a little bit more obtuse. Ladies, this is the fornix, Fornix, the fornix. Let me type that into the chat window. That's the fornix. Okay, the third practice question with the eyeball. What do you think that was? Okay, Pamela and Lindsay, both of you got it. Nicely done. The choroid. Very, very nice. That's exactly correct. And then the fourth practice image. What do you think this is? Okay. Does it help you? Okay, Pamela, it's the vestibulocochlear what? Vestibulocochlear what? Right, it's the vestibulocochlear nerve, which is a cranial nerve. It's the vestibulocochlear nerve. That's correct. Notice how it's coming out of the internal auditory meatus here, right? You see this little hole coming out of the internal auditory meatus. Okay, very nice. It seems to me, ladies, that you probably got 3 out of 4. which is not too bad. It's really not too bad. Okay, so good job on that. All right, so I'm going to stop sharing that and simply, is there anything that I can help you with at this time? What happens when your sclera keeps growing? Well, thank you, Lindsay. Have a good weekend too. Okay.